set of slides is on the influenza virus. And so we'll start with a little bit of classification. So here's a big classification scheme based on a couple different features that include the, mostly the phenotypic characteristics. So in this particular case, it's the nature of the genome, either minus or plus strand, and then the nature of the capsid, and so forth. And so in this particular case, if we follow this down, here's influenza virus here. These are single-strand RNA genome viruses that have a negative sense RNA. They're envelope viruses, but what's interesting about them even further is that they have a segmented genome, as I'll talk about, and then they fall into this particular family, Orthomyxoviridae. So here's another virus that we'll be talking about, HIV, and you can follow that through. These are plus-stranded RNA viruses. They have a diploid genome, meaning they have two copies of their RNA in the genome. They do have an envelope. They have a conical capsid, so it's shaped like a cone essentially. And they are in the family Retroviridae. And here's our microbe. And then the genus Influenza A, Influenza B, Influenza C, and Influenza D. So there's multiple genera. And just some features of these viruses. Influenza A virus is the major human pathogen, whereas B is a human pathogen, but rare. And we did see a lot of influenza B cases this past flu season. And influenza C is very rare, very mild, but does occur in other types of animals. And then, of course, influenza D is primarily an animal pathogen. So here's a picture of the virus, and you're looking at some of the features that I had mentioned. In this particular case, the segmented genome is illustrated here, and actually there's more pieces than this, but I'll show you a different picture. Two major spike proteins on the surface of the virus that give it its characteristic appearance, but also give it its classification scheme. And so in this particular case, you would see a typical influenza classification with these various components. So whether it's an A, B, or C virus, or A, B, C, or D virus, where its geographic origin is, a strain number, from a lab, the year of isolation, and then lastly the subtype. Subtype is a direct reference to the types of spike proteins that the virus has on its surface, what particular kind. And so there's two different types. There's hemagglutinin and neuraminidase. And so I'll talk about what these do, but you've probably all heard these H1N1, H3N2, and so forth. And so these are different kinds of hemagglutinin, for example. There's a variety of different hemagglutinins, and then there's a variety of different neuraminidase subtypes. And the influenza viruses can be characterized based on the particular type that they have. So going back in time, we can look at the 1918-1919 influenza pandemic as one of the more frightening pandemics of our time, much more deadly than SARS-CoV-2, COVID-19. But the newspapers were full of information, just as they are now. Most of it's crap, but anyway, in this particular case, there's possible mechanisms of spread, and then very startling headlines about the numbers of deaths in different locations and who's being infected, and then opportunists trying to come up with remedies. Hey, we sell bikes, and look at that, if you ride a bike you can avoid catching the flu. And then warning signs like we're seeing now, please wear your mask or whatever, and here it's spit spreads death. And then some different kinds of remedies, Zenol, some phenolic inhalant or something like that. I'm sure that's what that is, that you can inhale to kill flu viruses in your nasal passages. Who knows? But anyway, the point is that nothing really changes. I mean, all this stuff that was happening in 1918, 1919. We are seeing now, just in terms of the headlines and the potential cures and the warnings and the opportunism and so forth. Some of the features of the flu, the specific subtype was H1N1, so it was an influenza A H1N1. We still don't know really where it came from. They call it the Spanish flu because everybody wanted to believe that it was originated in Spain, but it just so happened that Spain was a neutral country at that time, and so they were very more open with their reporting of the pandemic, and as a result, they were more free with the descriptions of the outbreak, and so people just naturally wanted to blame them for it because they weren't hiding it like other countries were. Odds are it should be called the Kansas flu, Kansas pig farm flu, because there's a lot of evidence that it originated in a pig farm in Kansas. But of course, the United States won't cop to that blame. It's estimated that 50 million people died worldwide, but probably more, in the 100 million range, which is insane. 700,000 US deaths, about a 2.5% CFR, case fatality rate, overall. So again, CFR is a reference to how many people died as the numerator over the total number of infected. So you can imagine that's a really next to impossible number to really pin down. But especially if you don't have expansive testing going on, and that's one of the problems that we're having right now with SARS-CoV-2 is trying to come up with a CFR for that without really knowing the extent of the denominator. So typical seasonal flus have about CFR of about 0.1. And so the virus has, as I said, multiple genome segments, as I'll show you in another couple slides. And so individuals went to places like the Arctic and found graves of dead people that died of the flu, and dug those individuals up, and then were able to recover bits of flu genome, 1918-1919 flu genome from these corpses, and because obviously they're essentially buried in permafrost, so they're very well preserved. And because the goal of the research was to try to figure out why this particular flu was so deadly, and so they pieced the virus back together, which is which is frightening, and obviously it led to a lot of discussion amongst microbiologists as to whether or not to allow this research to even take place. And eventually it was said, okay, we got to do this in the name of science, but it is scary to think about resurrecting a virus like this. Anyway, they took some gene segments from the 1918 flu and made recombinant viruses and found them to be highly virulent in mice, causing massive cell damage in the lungs. And obviously upregulation of apoptosis genes in the lungs, meaning that the lungs were being forced to kill themselves. And so this is an interesting aspect of it, that the virus was unusually virulent and capable of causing massive cell damage. It had a very unusual specific death rate curve as well, and so that's pictured here. And so typical death rates look sort of like this, where there's a sort of a U-shaped curve. So this is specific death rate, and this is age range. And so what this is telling you is that in its typical flu, the very young and the very old are highly susceptible. But with the 1918 flu, it appears pretty clearly a W shape, which means that individuals in this middle age range, which are usually spared by the flu, actually were dying in high numbers. And of course, that's frightening, and for a variety of reasons. But not unsurprising, right? Because you have to remember what was happening. This is, World War I was going on right now, and so these individuals were packed into large ships and transferred across the ocean. They're in trenches. They're very close together. They're sick and stressed, and so it's not surprising that a lot of individuals in fighting age, were succumbing to this particular disease. The pandemic also came in 3 distinct waves. So here's the first wave, which was a bit of a blip, and everybody thought, phew, we're out of it. And then the second wave came, and it was far deadlier than the other 2 waves. And then there was a later third wave. And this is why we're hearing some of the SARS-CoV-2 fear about a second wave. I mean, we had a bit of a first wave for sure in certain parts of the country. But there is fears that in the fall we're going to have a second wave. But the difference here is pretty stark. The influenza viruses, as I'll show you, have a different way of mutating. And as a result, they're more likely to change significantly from here to here. And that's most likely probably what happened with influenza. The virus came on and then had some pretty significant mutations that individuals that weren't resistant to those mutations were very susceptible. Even individuals that got infected here could have theoretically got reinfected here. And we don't expect that with SARS-CoV-2 because the virus doesn't change so much. And as a result, people that were infected here in the spring will not be represented here in all likelihood. And so what happened when the individuals recreated the virus, the 1918 virus, using the resurrected RNA from corpses frozen in permafrost, they found that the normal tracheal cells looked like this, but then when they were infected with this particular strain of recombinant flu that contained some of the RNA from this old strain, there was almost a complete denuding of the tracheal cells. The microvilli were gone. And that led to, presumably leads to decreased clearance of mucus and increased colonization by bacteria. So mucus that's secreted here normally would move up the trachea into the larynx, into the mouth, and then you can spit it out or swallow it into your acidic stomach and kill the microbes. But without that mucus clearance, bacteria could build up more readily. And so the hypothesis is that so many people died of the 1918 flu actually due to bacterial pneumonia because of reduced ability to clear mucus. And so this is typical peak flu activity in a broad range of data sets. And so you can see that peak flu activity is typically February, but it's generally a 1, 2, 3, 4, 5, 6 month deal. But with most of the cases occurring in the later part of winter and early part of spring. WHO data showed that the global attack rate, odds of getting sick, is about 5 to 10% in adults and 20 to 30% in children. Illnesses can result in hospitalization and death, and we know that. Even though most people aren't very worried about the flu year after year, there's a lot of deaths. Worldwide, these typical annual epidemics cause about 3 to 5 million cases of severe illness and about 250 to 500,000 deaths. So that's why a lot of people were saying that, is this really more deadly is SARS-CoV-2, COVID-19, really more deadly than typical flu outbreaks? Last year, our last flu season, the estimate of flu deaths is in this range, and this is where we sit right now with SARS-CoV-2 after nearly 5 months of dealing with this. So we are kind of on pace to replicate a bad flu season, essentially. Obviously, we're not done with the COVID-19 pandemic, but that's where we're at right now. Anyway, like I said, after 5 months. Influenza is referred to as the silent killer, and the CDC estimates that the flu is involved in actually 15 times more deaths each year than is recorded on death certificates. And this is something that people were trying to say with COVID-19, that it's a silent killer and there's more people out there dying, but that's nothing unusual. There's nothing unusual between SARS-CoV-2 or COVID-19 and what we see each year with the flu. So here are the estimates for flu deaths, for example, and then these are the actual reported numbers. And so again, there's not a lot of incentive to report flu deaths on death certificates. Often what's reported is the bacterial pneumonia or something that they get. And so that's why these numbers, these red bars, are much higher. And the same thing is said about COVID-19. But the problem is a lot of people don't realize that influenza does the same exact thing in terms of predisposing you to bacterial infections, which are actually the cause of death. The difference is COVID-19 is a hyper-political infection right now, and so nobody wants to miss out on labeling something a COVID death. And so it's very unlikely that your bars are going to be like this, of missed COVID deaths. So again, that's an interesting side effect of infusing politics into public health. So here's a here's a overview of the symptoms of influenza, and again, they're wide ranging, just as a lot of people say with COVID-19. No difference. Influenza does all the same things. There's absolutely no difference. Even the fear of Kawasaki disease in children happens in influenza patients as well. But the difference is influenza is far deadlier for children than is COVID-19. The symptoms of influenza: runny, stuffy nose, sore throat, body aches. All of you have had, I'm sure, influenza, and so you know this: extreme tiredness and high fever, and headaches and so forth. So getting into the biology of the virus, there's a lot of really interesting features to the virus. The genome is arranged in 8 segments, as I talked about. So this segmented genome gives this virus a lot of advantages to many other kinds of virus, including SARS-CoV-2. So 8 segments and a common ribonuclear protein that lines up with each genome fragment to provide protection. So these nucleoproteins that you can see, these little purple spheres that are bound to each particular fragment. The nucleoproteins arrange each RNA into helical structure, and so again, there's a polymerase here at the tip, and then there's a helical RNA, and again, it's a single strand of minus strand RNA. But the way but one end of the RNA is bound here, and then it winds through helically, and then the other end is bound to the other end of the polymerase. So the nucleoprotein, RNA, and the polymerase are bound to the 5' and 3' end of the RNA, and so that arranges this in a circle. It looks pretty twisted right here, but if you unraveled that, it would be just a bunch of circles. And so again, you can see the types of features here. So here's the polymerase complex, as I already mentioned, the nuclear protein complex, the viral RNA that makes up each of these segments, the hemagglutinin, the neuraminidase, little nuclear export proteins, and then these extra little membrane proteins referred to as M2, which I'll describe. And then there's matrix proteins, this purple layer here, and then the lipid envelope, which is this layer here. So influenza receptor that the virus looks for is sialic acid that's bound to an integral membrane protein, and that's pictured here. So here's our integral membrane protein. These guys are found in high abundance in your lungs and respiratory tract, not surprising. This part right here is the sialic acid that's sticking off of these cells in your lungs and upper and respiratory tract. And then this little yellow dot here is galactose. So this is what the virus is looking for, this particular receptor here. Specifically, the hemagglutinin portion is looking for this sialic acid. They are located everywhere, not just in the lungs. They're their nose, throat, and lungs as well. And so they're even found in the brain, but the virus doesn't have the ability to easily get to the brain, so it definitely attacks whatever sialic acid it can find immediately, and that obviously is the respiratory tract. The virus enters through something called a fusion peptide, and so the virus's hemagglutinin binds to the sialic acid residues, as you see here, and then the virus is brought into an endosome. The endosome acidifies, so here you see the protons moving in. That acidification of the endosome ultimately allows the acids the protons to translocate across the virus through that M2 ion channel that I showed you previously, and that acidifies the internal core, and that changes this membrane fusion peptide in the that's associated with the hemagglutinin in such a way that it allows it to fuse the membrane and then spit its genetic information into the cytoplasm. So all the RNA and nuclear export proteins and polymerases and so forth and are spilled into the cytoplasm. And as you see here, the RNA fragments can then move into nuclear pores, and then those RNA fragments can be transcribed into plus RNA, and then the plus RNA can move back out and be translated into protein.